I'm Tom McNally, and this is my master's research project. It's all about how microbial diversity enables populations of bdelloid rotifers to subsist on inorganic ions. Now, I get a lot of questions. First of all, what is a bdelloid rotifer? Well, the B is silent, and bdelloid rotifers are microscopic animals. They're about 200 micrometers long, and they're down here in the protostomes. We have the ecdysozoans over here with the tardigrades and the arthropods. More on tardigrades in a bit. I want you to look over the spiralia, where branching off of the mollusks and the platyzoa like tapeworms. In the spiralia, we have a phylum called the Syndemata. And in the Syndemata, we have two major classes. We have the monogononts and we have the bdelloids. And the bdelloids have about 450 morphospecies. Why do I say morphospecies instead of just species? Well, that's because about 30 to 50 million years ago, the bdelloids lost their genes for meiosis and for sperm and for males and haven't looked back since. So they're all clones of each other, but despite this, they are still diversified. Really useful for research because you can just put one somewhere and go away for a little while and then come back and there's 100 genetically identical clones. The bdelloid that we've been using for our research is Adineta ricciae, which I will render here in a more whimsical style. Now, ricciae is really useful as a model because she's very easy to culture. You can pick her up quite easily with a pipette and put it in places and construct an environment. Really good for experimental work. They tend to live in liminal terrestrial environments, places like mosses and lichens. They end up in a lot of bird baths and mud patches and gutters. And they scooch around on all kinds of surfaces and they graze on microorganisms. They're pretty remarkable and somewhat famous for being able to withstand all kinds of punishment. So cold is no real problem to them. If you take all the oxygen away, they can sleep through that. And ionizing radiation, it seems any kind of mutation, they can quite readily repair. Really remarkable capabilities they have, which aren't yet fully explored. They can also withstand starvation. So you put them somewhere without much food, and they can just wait it out and recover when food becomes available. And they can now scooch around and start looking for microorganisms again. And part of all this extreme resistance to stressors comes from their ability to undergo anhydrobiosis, which you may have heard about because it's something tardigrades do as well. Tardigrades also live in limnoterrestrial environments. They tend to be found in the same sorts of places as bdelloids. With anhydrobiosis, the animal can detect when their environment is drying out, when the water level's coming low. They can scrunch their heads into their chests and they can scooch their tails up into their tummies and they can form a sort of barrel-shaped little dried out form. If they're stuck to a piece of debris, they can sail around on the wind in what's in a passive dispersal. They can now travel to sort of lush environments that might be full of food, but then also they can wind up in pretty inhospitable places, which is where all their resistance to stressors comes in handy. They've gone from the Arctic to the Antarctic. In fact, bdelloid rotifers are found in every single terrestrial freshwater source on the planet. And these extreme ephemeral environments are really interesting because they all wind up in a place that's bare rock. There's no plants or algae or anything converting inorganic ions into food, but still they're able to gather up enough nutrients which they can use to make more bdelloids and disperse again, so long as there's no competition. I think that's pretty interesting and something I wanted to look at. So, can they just grow a population? Can that grow from just inorganic inputs? And if so, which inorganic ions are essential to that process? So to model a kind of extreme Antarctic patch of bare rock, we took a founding rotifer, and if you wash it in just molecular pure water to wash up any kind of organic carryover and put it on a bare petri dish, just in a solution of ultra pure water, that's just molecular water with NOP solution, which is a botanical growth medium that I'm going to talk about quite a bit, so I'll define it for you. It's got calcium nitrate, it has potassium chloride, it has potassium phosphate and magnesium sulphate. These are all the kinds of these are salts that dissolve into the kinds of ions that you kind of need to build basic organic molecules. So what happens if you... So there's no food in here, there's no bacteria except what they've brought with them. What happens to a Adineta ricciae if you put her in there? Well, she's able to increase her biomass from something. Here are the results to show that I'm not lying. Each of these groups had 11 replicates or 11 dishes. Over here we got the lettuce, that's the positive control, that's a rich supply of food. And then we have these 3 different NOP solutions, so that's with the 4 inorganic salts which dissolve into the ions they need. There's pretty much no difference between these different volumes of NOPs, or this older formulation of NOPs I used. But then if you remove any of the NOP salts, it doesn't work. It's no better than control. And the control here was the ultra pure water. So this shows that they're not deriving nutrients from the plastic of the dish. There's no photosynthesis going on. If they're in ultra pure water, they can't build a population out of the environment. There is a caveat to this. If you take a founding egg and you wash it in bleach, that removes the microbial community from the exterior and interior of the bdelloid, and for whatever reason they're not able to increase their biomass from there. They're not able to grow up a population. Whereas if you just wash them in water and transplant them to a new place, because you haven't killed off their microbes, that's the sort of key. The microbes they bring with them is what they're going to eat. So, from this experiment, can Adineta ricciae grow solely with inorganic inputs? They can, so long as they have their microbial community, and the ions they need are the kinds of ions that you'd expect. In fact, these the salts that we used, one of them was a little less pure than the others, so there would have been trace amounts. It was like 95% potassium chloride. There would have been trace amounts of iron and sodium in there as well, which makes sense. It would be pretty weird if they were able to build a whole animal body without those sorts of essential ions. So we have a picture here where you have these ions, and they are enough to feed a microbial community. The microbial community can make more microbes, and if there is a rotifer founder, that founder is able to eat the microbes and build up a population. So the biomass is coming from the microbial community. So, if we're to assume this is a synotrophic community where some microbes are producing metabolites and byproducts that are feeding the others, and that they are interacting in all kinds of ways, taking the ions, building up this community in order to now produce this community of rotifers, I want to know what's in that black box. Well, there was a handy tool in order to take a look at what's inside that box, which is the DNA evidence. Can we use that DNA evidence to identify all these metabolic pathways which the Adineta ricciae microbial community has? The pathways, just to make it clear, which they're using in order to turn these inorganic ions into organic molecules, the kinds which you can build bacterial microbial bodies out of. And which of those pathways are involved in building organic molecules, and which of those pathways are involved in generating energy for respiration. So, I had the metagenome of the Adineta ricciae community, because the Adineta ricciae genome itself was sequenced here in Imperial. Now, by taking an unfiltered version of that metagenome, which had all the bacteria, microbes, and stuff left in it, I was able to run that through a KEGG mapper to reconstruct the pathways, and what we have here is a map of all the sequences from all the little microbes and species which were not Adineta ricciae. So all the sequences that make genes and enzymes that are involved in these specific metabolic pathways. So the yellow blocks here, they are hits. They are sequences that were actually present. So those are genes which we presume are being expressed to make these to make these enzymes which are involved in these pathways. The first place I looked was in the methane metabolism, which you can see here, and unfortunately there was not the thing I expected to find in the methane metabolism. I was looking for methanogenesis or methanotrophy, and there's no evidence to say that that's going on in the A. ricciae metagenome we looked at from the lab species. But a bit more encouragement with the sulfur pathway here. The assimilatory sulfate reduction pathway is more or less complete. So that means that in this community there are the enzymes to turn sulfate into sulfide. And once you've turned sulfate into sulfide, you can build that into amino acids like cysteine and methionine that contain sulfur. No reason to believe that there's a dissimilatory sulfate reduction and oxidation pathway, so they're not using sulfate for respiration. They're not using that to generate energy, which is a shame because they've got access to the sulfate in the NOP solution. The nitrogen metabolism was much more promising because here you can see we've got nitrate, which can be turned into nitrite. Bit of a gap there between nitrate and nitrite. But once you've got nitrite, convert it into ammonia, and once you've got ammonia, there's actually several enzymes that are available for that. It can be converted into glutamate, and that once you've converted into glutamate, you can. That's an amino acid. You can also convert it into arginine. So that's protein synthesis over there. But then also there's this really interesting enzyme that's present, which can turn glutamate back into ammonia and into 2-oxoglutarate down here. And 2-oxoglutarate is an intermediate in the citrate cycle, so that's something that can be used for respiration. This is the enzyme that's involved in that. That's glutamate dehydrogenase, and that's a reversible reaction. So there's enough enzymes in here to make good use out of nitrate. So, is there DNA evidence? Yes, you can construct pathways from this unfiltered metagenomic data. It's not a smoking gun, it's definitely a direction to look in. It's good to know that methanogenesis is not terribly supported at a first look, but then a nitrogen metabolism is. So maybe we could look for ammonia. Can these pathways generate energy and build organic molecules? Well, yes. It looks like there are the enzymes in there to build organic molecules from nitrate and sulfate and to generate energy from nitrate. Looks like there are these lithotrophic processes that the rotifers can take advantage of, either by eating the microbes directly or eating the things that subsist on the lithotropes themselves. So, let's go back to these bleached rotifers, because there's something interesting you can do with them. So the rotifer egg is bleached, removing its microbial community, so you have this azenic, no microbe rotifer, and their microbial community can then be reconstructed by adding in isolates of single species. And there's actually a species of Pseudomonas that can support a quite slow growing but functional ricciae community. So this is a monozenic rotifer with just one species of bacteria that it just lives in and eats constantly. However, this monozenic community, if they're just raised on NOPs, if they're in ultra pure water and the NOP solution, they can't produce whatever metabolites are necessary in order to support this rotifer growth that we've seen in rotifers with a full microbial community. So, can you then reintroduce the full community and restore this ability to make use of these ions? Early experiments showed yes, so I wanted to take that to an interesting place, because there's a question that's been arising here. Is this effect of being able to subsist of inorganic ions, is this something peculiar to the Adineta ricciae that we've been working with in the lab, that's been just passed from petri dish to petri dish along with whatever microbes that came with it for 20 years, or is this actually ecologically significant? Are the rotifers just packing up their microbes and taking them with them on their journeys through the air to new lands, and are they kind of unpacking them like a picnic? Or is it more the case that wherever they go, if they land somewhere, they have a reservation? So here's the community inoculation experiment design. We have here 17 different microbial communities, these lab communities over here, There's a monoxenic lab community, a full lab community, and a lab community that's just been raised on the NOP solution, those 4 salts. Then we have all the samples from the natural community. And they came from these Q. roba oak trees that grow on Silwood campus. Here they are. Here's the full tree. And what I did is I sampled mosses, either from the ground level of these trees or from 1.5 meters up the tree. Now, previous research has shown that the rotifers are far more at home, and a greater proportion of them to other animals, up the tree, because at 1.5 meters up the trunk of the tree, the moss is more subject to the air, more subject to the sun, it's more exposed, it dries out, and everything desiccates, and the rotifers are very happy with that. And it seems that they shed their parasites and other things that may cause them harm. Whereas down on the ground level, by the soil, there's a much greater diversity of microbes and more likely to be negative interactions in there. So, if you take the lab community with its full, what the rotifer's been growing in for 20 years, and you give that to these monoxenic rotifers, does that help them out? Or is it better if they get, their greens from a piece of moss from a tree taken from outside? And is there any difference between the different samples of natural communities, or is there different levels between heights, or both? So, here are the results. You can see right away that the natural communities win out. So, whether it's from high or low, the ground samples are much more likely to support a higher population of rotifer growth than the lab communities. And all 3 lab communities aren't terribly good as a group. The most surprising thing was that the full lab community over here was expected to be a positive control. It was expected that that would be a proven way to rescue the rotifer growth, but actually it seems that there's something in the lab community that's harmful to them. It's a negative interaction, actually suppresses their growth. The NOPS community and the monoxenic community are about level-headed, so we think that's probably the baseline. Next up, looked at tree to tree, and it seems that there's actually no difference between each tree. That makes sense. The trees are all in roughly the same place, checked for things like cardinal heading, but no, as far as the rotifers are concerned for supporting their growth, any tree is a good tree. So you see that there is this difference between high and low. This is one tree that's been sampled 3 times, and you can see it quite starkly here. The ground samples, interestingly enough, are much better for the rotifers as a food source than the ones from higher up. That's a bit surprising, not what we expected. So, does an inoculation with a natural community improve the growth of rotifers? Better than an inoculation from a lab community? Yes, actually. The natural communities have a larger effect than the lab communities, and some of the lab communities can be harmful. Of the natural communities, there doesn't seem to be much difference between trees, but ground level communities have a larger effect and higher variance between them than the communities taken from 1.5 meters up. Now, I'd like to return for a moment to the remarkable abilities of rotifers to withstand these stressors. And it hasn't escaped the notice of people interested in astrobiology. In fact, the bdelloids have been considered for a couple of space missions. An interesting one was the Moon Next ESA lander that would have deposited a little sealed environment of rotifers on the moon. Of course, now we know that the moon is peppered with tardigrades by accident, so I'm sure we'll hear a bit more about this kind of thing. But I'm interested in Mars and how they'd get on if they found themselves on the surface of Mars, on that bare, exposed rock with harsh sunlight, low air pressures, etc. So I exposed them to the surface of Mars, which in this case was in Milton Keynes. Here is Edmund. It's a decompression chamber, the kind that you'd go in if you'd had a bad time diving, and I packed them in dry ice to bring the temperature down to minus 78 degrees, and shut the chamber and exposed them to different lengths of time of Martian conditions. The hypothesis was, is the survival rate of these rotifers that have been desiccated and put on these little pieces of filter paper and put in this Martian chamber, are they different to a control group which had not gone in the chamber, which had just been desiccated and taken to Milton Keynes and taken back rehydrated and counted. So, on counting, I let them rehydrate for 3 hours, and it turns out that there's actually, statistically, no difference between any of these groups. The control group didn't go in the chamber. This one went in the chamber for 10 minutes at room temperature. These two went in the chamber for 10 minutes and 40 minutes, packed in dry ice. It didn't matter. The survival rates between them were pretty much the same. So we can say that no, there's no significant difference between the control and up to 40 minutes of exposure to the gas mix and the air pressure on the surface of Mars. Unfortunately, the UV lamp wasn't working the day I was there, so we don't have radiation in the mix. What I'd like to do is to build up to a full Martian day of exposure. So, our conclusions are an Adineta ricciae population can grow solely with inorganic inputs, as long as it has a microbial community. The ions it needs are pretty straightforward, and it seems that they need all of the ones we've tried. We can access the potential metabolic pathways of what this microbial community may be doing. We can see that they can build up organic molecules from nitrate or sulphate, and that they may be generating energy from nitrate. We also know that an inoculation from a natural community improves the growth of rotifers in an inorganic ion solution, that NOP solution, better than one from a lab community, which they've been sitting in for 20 years. However, in natural communities, there doesn't seem to be difference from where you get that natural community from, but there is a difference in whether you get it from the ground level or from 1.5 meters up the tree, which is a proxy for diversity and a proxy for desiccation. And finally, if you put desiccated rotifers on the surface of Mars for up to 40 minutes, they're just as likely to be rehydrated and to come back into motion and movement than ones that hadn't done that. So the takeaway lesson seems to be that wherever you are on Earth or beyond, the bdelloid rotifers, wherever they go, there is a table waiting for them. Thank you very much for listening. I want to give a big thank you to my supervisor, Chris Wilson, as well as Miranda Burke and Joel Elkin, the students whose work I followed, and personally to Patricia Finnegan, Paul McNally and Stells Dessoy. Thank you very much.